Ladies and gentlemen, it gives me great pleasure to welcome you to the Advances in COVID Therapeutics panel at the Sachs Associates 8th Health Tech Investment Forum Digital Conference. On our panel today, we have, I'm sure you'll agree, a group of highly esteemed individuals, each with significant experience in developing therapeutics for the treatment of COVID. Therefore, with no further ado, it gives me great pleasure to introduce the panel. With me today, we have Sander van Deventer, operating partner at the European venture fund Forbion, who will be representing investee company Covercept. We also have Cameron Durrant, chairman and CEO of Humanigen, Richard Marsden, CEO Cinegen Research, and Torsten Mummenbrauer, CEO of ExeVir Bio. Unfortunately, due to unforeseen circumstances, my panel co-chair, Raoul Mova, will not be able to join us today. We send him our best regards. Before we start the panel discussions, though, I'd like to ask each member to briefly introduce their companies and COVID therapeutic approaches to you all. I'll start by welcoming Richard Marsden, CEO of Cinegen, to the floor. Over to you, Richard.
Thank you very much. Thank you very much, Max. I'm Richard Marsden. I'm CEO of Cinegen, and we are developing an inhaled form of interferon beta that we deliver directly to the lungs of patients with COVID-19 illness. So interferon beta is a protein that we all make when we get viral infections. It triggers many antiviral pathways, and it's particularly interesting in the context of COVID because this virus is a master at suppressing the production of this virus by cells when they are infected. And if cells don't make interferon beta, the fantastic pathways which are in place to defend ourselves against viruses and stop replication of the virus will be compromised. So we are delivering this drug directly into the lungs of patients at their moment of need, and that we define as being hospitalized due to lower respiratory tract illness. We conducted a clinical trial that was concluded in 2020. That produced very positive data, very encouraging data, and we rolled straight into a phase 3 trial that started earlier this year and is progressing very well with potential to deliver data in the second half of this year. I think the other thing to mention is that in the whole COVID treatment landscape right now, we have a cluster of therapies, mainly powerful anti-inflammatory drugs, which are being delivered to critically ill patients who are on ventilators. We don't see a near-term use for our drug in that setting. We may get to it very cautiously and carefully one day. And we also see a lot of activity now in development and progression of drugs directed towards patients as soon as they are diagnosed as SARS-CoV-2 positive. It's possible that our drug may have a place in that setting, but also last year we found that giving the drug to absolutely everyone was well tolerated, and patients found it relatively easy to use the drug in that setting. And in that setting, we deliver the drug and device directly to the patient's doorstep and coach them how to use the device and drug through Zoom calls and YouTube videos. That's perfectly feasible, but we see the moment of needing to deliver this drug into patients' lungs at the time when that human has started to lose the battle with the virus in their lungs. And that's the setting where we're generating the strongest data at the moment. Obviously, if we are progressing very well with our phase 3 trial, we need to prepare for launch next year, potentially under an EUA. And to support that, we have invested significantly in our manufacturing setup and also in distribution and in-market support. Because we have to be prepared for a positive trial result, and we have to be prepared that COVID remains a serious illness next year, and we need to be able to meet market needs in terms of drug and device availability, but also in market support.
Great, Richard, thank you very much. Now gives me great pleasure to introduce Cameron Durrant, CEO of Humanigen. Cameron, welcome to the floor. I believe you've got some slides as well, so if we could start those slides as well.
Thank you very much for inviting me. I have 4 slides as instructed by Sachs, not counting the first 2. So here we are. Humanigen is a publicly traded biotech company. We are developing lenzilimab. Lenzilimab neutralizes GM-CSF, which has been shown in multiple studies, including the largest conducted to date by the UK government that was published in Science Immunology in March of this year. We completed a phase 3 520 patient study, which we refer to as LiveAir. And I'm showing you the top line results. We hit the primary endpoint. Failure to achieve survival without ventilation. We sometimes refer to that as SWOV, survival without ventilation. That was defined as mortality or requiring invasive mechanical ventilation. And just to be clear, we are focused on hospitalized patients. This is not an outpatient monoclonal antibody. Neither is this targeted for patients who have already undergone invasive mechanical ventilation. And you can see from the histogram there that the proportion of people in the lenzilimab arm versus placebo was different. It hit statistical significance 0.0403 with a hazard ratio of 1.54. And again, to be clear, placebo does not mean nothing. This was standard of care as deemed by the physician. 94% of the patients in this study received steroids, 72% received remdesivir, and 69% had both, as well as potentially other therapies. So this benefit from lenzilimab was over and above what is seen as a benefit with other standard of care therapies. Next slide, please. We also looked at an exploratory endpoint, which was baseline C-reactive protein levels. 150 has been established in many publications as a cutoff, much above 150, and the patient is really very, very unwell. So in a normal situation, you or I may have a CRP in the low single digits. When we looked at this biomarker of serious inflammation, you can see on the left, this is similar to the SWOV analysis I showed you just now, but in the CRP less than 150 range. So these are people who are a little earlier in the hyperinflammatory phase. Typically, they'll still require supplementary oxygen, or they'll be less than 94% on room air. And you can see the difference is even greater, and the p-value is 0.0009 with a hazard ratio of 2.54 versus 1.54 in the prior analysis. We also looked at Black and African American patients. And when you look at the same segment, you can see a remarkable difference. Black and African American patients are hyper vulnerable to COVID. They're hospitalized at 3 times the rate of people outside the community, and they die at twice the rate. They represent 13% of the US population, yet 23% of the deaths. And you can see a very significant difference. And again, it hits a p-value of less than 0.05. Next slide. The safety profile. Essentially, lenzilimab has proven to be comparable to placebo in terms of safety and tolerability, and that's consistent with other studies that we've conducted with lenzilimab in other settings. So I'm going to finish up my time by saying that we believe that there needs to be a greater balance with respect to the investment in vaccines and therapeutics. So it's not either or, it's both. They go hand in hand for people who are unable to take, don't wish to take a vaccine, can't access it, have reasons, medical reasons or other reasons why they choose not to take it, or in fact the effect wanes after time, and that appears to be where the evidence is tracking, then if those patients are hospitalized and they deteriorate, they need more and better therapeutics, and those therapeutics need to be variant agnostic. And I'm sure we'll be talking about the emergence of variants later on this panel. Thank you.
Great, thank you, Cameron. That's really good to see that data, and obviously wish you all the best with prosecuting that data through regulatory pathways. Now it gives me great pleasure to introduce Torsten Mummenbrauer, CEO of ExeVir, to tell us more about ExeVir and what you're up to in the COVID space. Thanks.
Thanks, Max, and hello to everybody. ExeVir is a clinical stage biotech company developing single-domain antibodies, which are also known as nanobodies. We want to build an infectious disease pipeline, and we have a lead candidate in COVID-19. The lead, ExeVir-011, is a broadly neutralizing antibody, and to say we are quite proud that we brought it from the bench, from the lab to patient in less than a year. Nanobodies are a special class of small antibodies, just the VHH fragment derived from camelids, in our case from a llama. And llama winter had been vaccinated with the spike protein of SARS-CoV-1 by the scientists of the VIB in Belgium, Flanders, already 3-4 years ago. And as soon as the SARS-CoV-2 sequence became public, they did a screen and found 100% cross-reactive binders. And so that was, I think, the birth of our lead candidate. Nanobodies are very small, I mentioned that before, and so that gives us the advantage of some very special features. So in our case, we have fused the VHH to an FC. So we have a homodimer, 2 VHHs on one FC. We look like a monoclonal antibody, but we are still roughly half the size of a monoclonal. It's roughly 80 kilodalton, which gives us a much better tissue penetration, important feature. As I've said, we originally developed it against SARS-CoV-1, so we are targeting a very special epitope, a hidden epitope on the spike protein. It's only addressable, accessible due to the small size of the nanobody, and it's highly conserved over all sarbecoviruses, which also speaks for the importance of the epitope for the virus. We have no data which shows any change, modification of potency of our molecule against any of the variants. So we still high potency and the same level of neutralization against all VOX and also against the variants which are under consideration. One additional important differentiation factor is we have silenced the FC of the molecule. So we want to have the maximum safety of this candidate and want to avoid any extravasation of the disease in infected people. We are clinical stage. We have a phase 1 in healthy subjects running, or we have infused the first cohort, very low dose, 250 milligrams. Second cohort will be done this week. No safety signals so far. And we have a 1B open in Europe. We have the first sentinel patients in the study, and we have 5 sites now open in Europe. Interim data should be available for dose range decision mid-November. We will then immediately follow up with a phase 2 registration trial design, placebo-controlled randomized international study, including US, Mexico, Brazil, South Africa. Readout is planned for March. We want to be ready with an MAA filing, mid next year. And we target temporary conditional approval in Europe for next year. All that only makes sense if you also have a product available. And so we have completed the tech transfer to a European-based CDMO. We have booked 7 slots for manufacturing starting this winter. And so the first one will prove the tech transfer. Then we have 3 consistency runs, which are important for the filing. We discussed rolling review with the EMA and hope to get the data in then over Q3 next year. And that means we could get conditional approval, hopefully in the winter 22, which would allow us to ship the first antibodies to patient then in roughly 1.5 years from now and make ExeVir a commercial stage company. All of that costs a lot of money. And so we've just reached out to the venture community again, and we target to raise a rough 100 million over the next 12 months. We want to do that in a two-step process, so roughly 50 million under a Series B, hopefully before year end or beginning next year. And the additional 50 plus million, we'll have a look at the public market next year or consider also Series C. Thank you.
Great, Torsten, thank you very much, and good luck with the process and the progress you're clearly making and plan to make. That gives our final. I want to introduce our final speaker, which is Sander van Deventer from Forbion, who is going to be talking about Covercept, one of their portfolio companies. So, Sander, over to you.
Yeah, thank you very much. Covercept develops a small molecule that was discovered at the University of California, San Diego by 3 scientists, Philip Gorge, Jeff Esco, and Sam Tsimikas. The small molecule addresses a host mechanism, which is very well characterized. And the result is threefold. There is a very effective prevention of viral binding through heparin sulfate proteoglycans that actually has been published, those data. In addition, there is a potent inhibition of viral replication in human cell lines and also in 3 independently executed hamster models. And the viral replication inhibition is far more potent than remdesivir. We're talking about at least 3 logs better. And then in the hamster, it was also found that there is a profound anti-inflammatory effect, and in particular, an anti-fibrotic effect. So if you look at the genes that are involved in fibrosis, then most of them are actually potently inhibited. We also believe, and this is interesting, that because it targets a host mechanism, that these results can be generalized to other RNA viruses. And in fact, data now have confirmed this for Zika, chikungunya, dengue, and SINBIS. And we also believe that SARS and MERS will be inhibited. And for West Nile and Ebola, this is under investigation. The drug is safe, can be orally administered once a day. Has a very nice pharmacokinetics. It actually in animal models accumulates quite significantly in lungs. A large-scale GMP manufacturing process has been established, and the company is now conducting a phase 2 clinical study in Brazil with a primary endpoint of viral clearance, which is expected to read out later this year in Q4. So to summarize the target profile, it is believed that this drug could work in early stages of COVID or even in preventing COVID, but also in late stages, particularly with a look on the anti-inflammatory and anti-fibrotic effects. And then the drug can be easily stockpiled and is believed to potentially be effective in future pandemics as far as they can be, as far as they depend or are caused by RNA viruses. So that's Covercept.
Great, Sander, thanks very much. So we'll now open up to sort of the questions to the panel, and I mean, clearly, I think from your presentations, you've all demonstrated that some interesting clinical data, some small molecule approaches, some antibody approaches, some biological approaches. So quite a broad range. I wonder, and perhaps we'll start with Sander. What have been the key bottlenecks that have arisen in terms of developing specific therapeutics for COVID? Because clearly the vaccines have maybe grabbed a lead over the therapeutic side. And as I think Cameron said, we need a balance of both.
Yeah, so I totally agree with that. I think the passive immunization side and of course the vaccination side have developed very rapidly and in a certain way have been very effective. But I think going forward, of course, I think the one thing to consider is that COVID is not going away. So I think this is going to stay with us. So now we need to have longer term plans and also to have a rapid response when a patient gets infected. So we need therapeutics basically. And when it comes to therapeutics, you can think about therapeutics that interfere with inflammatory responses or interfere with viral replication. I think the Covercept drug actually does both, and I think going forward these will become the mainstay of future early responses to outbreaks of COVID-19 and maybe MERS and SARS.
But what has stopped, the therapeutic companies developing products as rapidly as the vaccines? So, I mean, what's been the bottleneck for Covercept to move forward? Is it kind of just, obviously in the vaccines, what was seen as a very long process has been shrunk down to less than 12 months. And in the therapeutic area, we've had repositioning of molecules, but we haven't had specifically designed therapeutics. So are there any specific areas which, have led to that, do you think?
I can briefly address that, and then others no doubt will have to say something about it as well. But I think we need to remember that the vaccines had an incredible head start because of all development of the development of cancer vaccination programs and all that. So if you look at the RNA vaccines and particularly also the adenoviral vaccine technology, it was already ready for prime time because of all that previous work. So there definitely in the vaccine area was a head start. And I would say if we would not have spent all those billions on and all those years developing these technologies for cancer, I think we would have never been where we are now. So vaccines had a head start, and that clearly is not the case for small molecules. Okay,
and maybe then opening that up to Torsten, maybe because you obviously had a molecule that had already shown efficacy in SARS-CoV-1, so you almost were there. Is it funding then that's been the difference between the vaccines? There's been a lot of money. I think Cameron, you mentioned this as well. There's been much more money put towards the vaccines than towards the therapeutics. You had your SARS-CoV-1 molecule already.
Absolutely. I think I agree with Sander on the head start of the vaccines. I think the world didn't consider efficacious treatment in infectious disease before, I think, on the right level. There was an enormous focus on vaccines. And if you think about organizations like CEPI, they were just funding vaccine development with billions of dollars and nothing into therapeutics. And I think people are realizing now, or it took them a while to realize, that therapeutics are a very important component also in pandemic preparedness. If you want to ring fence an outbreak and you need to wait for the upset of the immune system after a vaccine, that takes time. And therapeutic efficacy is there immediately. And we have seen that. I think we wanted to raise additional money fall, early winter last year. And there, the focus was really 100% on vaccines. Politicians were telling everybody everywhere that COVID-19 is over anyway, because everybody will be vaccinated in the next couple of months. And so the interest on therapeutic development was very, very low at that stage. So we were convinced it will be needed. We continued, we found ways to continue. But at that stage, money clearly was a bottleneck. And these days, I have to say, to a bit more down-to-earth issues, I would say, everybody is facing the limits on raw materials. And I think that impacts vaccines as well as therapeutics. And it's simple things like media to grow your cells for production, or to get resins and columns for the purification, the downstream process. And in the meantime, you have lead times for certain products, which are essential, between 6 and 9 months. And specifically for a small company, if you tech transfer and you need to establish your process somewhere, your established CMC process, which you can't change anymore, that simply leads to delays. And in our specific case, it led to a delay where we always said what should not happen. We really missed this infectious window in Europe over summer. So we have started our phase 1B. And then, I think good for Europe, the patients was not available for a clinical trial anymore. And so that's why we have started a phase 1A in healthy subjects, to be sure that we will be able to find the right dose. And so it's a complex situation during a pandemic where I would say multiple issues are blocking you.
Okay. Great. I know, Cameron, you have got some issues you brought forward already, so
yes.
Interested to hear your thoughts on it as well.
Yeah, in terms of bottlenecks, I think that there are at least 3 major areas that I think about. One is government policy. This is a global situation. It's been politicized. And so government policy appears to be informed by the prevailing political machine in that country. And one of the issues is that the vaccine doesn't carry a passport, doesn't care what country you're in. And so we need to have a much more thoughtful global approach. And clearly there are a number of institutions that could take the lead for that. And we've seen countries retreat to me first with respect to purchase of vaccines and potentially therapeutics. So that's one big area. Like Sandra mentioned, this is here to stay. This virus is going to be endemic, right? We have never eradicated a virus that has an animal reservoir, ever. And it's just not going to go away. So this, in some way, shape, or form, and human beings are sharing the planet for the foreseeable future, if not forever. The second, I think, is clinical regulatory. So some regulatory bodies have been more open-minded, perhaps more efficient, responsive, and helpful. It does help to be a big company. I don't think anybody on this panel represents a big company. So it's a shame that we didn't have our colleague from Big Pharma join to offer their perspective. But those things matter, and that influence can help. Some of the other piece related to clinical regulatory is there have been examples of therapies and other interventions that have soaked up many patients, even when there's been evidence, hard evidence that it doesn't work. One example would be convalescent plasma. I'm not sure how many more studies need to be published that show it doesn't work before people stop enrolling people in studies. So there's a funneling away of potentially available patients. And then the third and final area is manufacturing. And I think Torsten touched on this, but there's a global shortage of media, resins, filters, columns, and so on, as well as downstream. So I understand there's almost a 2-year backlog for glass. So if your product happens to need glass vials, which most of our products do, then unless you've got that already booked and paid for, you're not going to get it.
Wow.
And some of the smaller companies, not just small companies, but some of the therapeutics have been bounced for vaccines. So that's why there's kind of this frustration that I'm expressing, I guess, around the myopic view that vaccines will solve everything. And we now have the booster debate. And perhaps I will shut up in a minute. The way I think about this is a metaphor where you have people drowning in the ocean. And some of those people have got 2 life jackets. The majority of the people globally have no life jackets. And the life jacket manufacturers are saying, hey, have a 3rd life jacket, or maybe a 4th. Variants of concern are primarily going to come from people who are unvaccinated. So unless we have a global response, is it better to offer at least just even 1 vaccination to as many people globally as possible than 2 or 3 or 4 or 5 in the more developed nations? Discuss.
No, I think that's a very good point. Richard, I'm going to turn to you on this topic of speed, because I know you had your data very early, I think last summer, and it was looked pretty compelling on the surface, and you had a good safety database behind the molecule from previous studies in different indications. So what have been the bottlenecks having had that original early data to getting your product onto the market?
Yeah, I don't feel we've had any particular bottlenecks that I'd single out or say anything different to other people on the panel. I would go back a step and sort of endorse what they're saying. I remember going to sort of pandemic preparedness conferences, of which there are usually 2 or 3 a year, going back 5 or 10 years. And at those conferences, there are a lot of people, you know, in terms of the agenda, a lot of it was devoted to vigilance, trying to spot the new virus, work out how to characterize it, and then be in position to study its spread. And then there was a lot of the agenda set aside for vaccine development and scale up. And I think we've all benefited from how quickly those vaccines did come into the marketplace. I mean, 11 months after we kind of all first learnt about the virus, which is phenomenal. And next time around, it might be 8 months. But that doesn't mean to say that we don't need therapies. We need therapies up until the vaccine comes along. If we start to think about the next big bad virus event, and it might be another coronavirus, it might be the influenza, it might be a adenovirus, or it might be a virus we don't even have a name for yet. So I think, and as you start to think about this pandemic, if you go back 6 months and things like that, people, I think, genuinely felt that the vaccines were going to provide a sustained, phenomenal response, because the efficacy is better than I think anyone would have predicted going back a year. And what we're seeing is some evidence of some waning of efficacy, and we're always kind of one variant away from going back to square one or square 0.5 or something like that. So the need for therapies is immense. And, even with these things in place now, people are still ending up in hospital in thousands of patients per day around the world. And that, we don't understand all of the biology as to why this is happening, but the problem exists and we do need these therapies. We've had tremendous support from regulatory agencies and also clinical trial networks around the world have really pulled together to help us. But I do know, because we've made some senior hires lately, that if you're not working in COVID space in our industry, it's hard enough in our space in COVID for all the reasons and the war stories that I'm hearing now from fellow panelists. But if you're not in COVID, it's nigh on impossible to make progress, whether you're trying to source materials for manufacturing, accessing CROs, or even engaging with agencies and governments. So I think the really tricky thing for being ready for these events is trying to predict which therapeutic is going to be the one that you want to accelerate. So vaccine technology, you can be prepared, finger on button, ready for a bad virus coming along. Antibody technology arguably has seen how it can be very beneficial. Trying to predict which of the therapeutics is going to be the one you want to have accelerated is very difficult. I think we will all benefit off the back of this pandemic, because there will be drugs which have bubbled up through all the clinical trials, which will present themselves as really valuable therapeutics for COVID. But if they have a broad spectrum profile, then there'll be the sort of drug that I'd hope governments would stockpile ready for the next one.
And I think what I'm hearing from all panelists here is that their therapies are potentially more broad-based than purely just SARS-CoV-2. So there's some hope there, and that's down to a later question. But maybe just briefly, I'd like the panel to touch on, early treatment or late treatment. This is something that's interested me in particular. Is is it important to get in there early, and then you've got a different safety profile issues that you have. So if you're in a community setting or a hospital setting, if you had to have the ideal therapeutic, is it one or the other, or do you need both? And I'll open it up first, Torsten. Just maybe let's do this briefly. Sort of where do you see the ideal therapy? Early community setting, or more just let's deal with the severe patients as they come in to a hospital?
I would address it not from the ideal spot there, but from the medical need. And we believe that the hospitalized patients have the highest medical need. You want to avoid severe disease, ICU, and all the other consequences. And I heard Cameron saying that convalescent plasma antibodies might not work. We believe that our nanobody hopefully is different. We have promising data, and we are addressing exactly this patient group. So we go in hospitalized patient, as I said in the intro, so we have silenced the FC to increase the safety profile of the molecule, and we want to address there the highest need. And we believe that there will continue to be the issue. I think in the next step, we want to go into the immunocompromised, and there that's the highest vulnerable population where vaccines, I'd say by definition, can't work. You do not have the appropriate immune response. And same is the truth for elderly people. I believe immune senescence is a fact. And so if the virus stays with us, you will continue to have disease and you will require therapeutics. And I have my doubts that this approaches to go earlier and earlier and try to prevent post-exposure, something like that. It's extremely expensive, that the healthcare system will be able to address that. And so I believe we are facing a situation where real efficacious treatment is required at the stage of hospitalization. Okay, so hospitalization is a clear call. Sando, are you in agreement with that, or do you want an early stage community product?
Yeah, it depends. So I would say it depends. Early stage product would be very helpful when you have very low pre-existing immunity. So, we do have 2 other coronaviruses in the circulation, which are common colds. And they are common colds because we do have pre-existing immunity. They could be very dangerous if they would be introduced in a completely, naive population. So it depends. If you have something that works prophylactically, then it would be a great thing. Let's say when new SARS or MERS or novel COVID or whatever variant would emerge, to which we do not have immunity. I do believe, on the other hand, also that there is a need for therapeutics, particularly targeting host mechanisms at later stages. And the reason, of course, is what we discussed, the elderly, the immune compromised patients, but also, I would say, and this is something we still don't know yet, the late stage effects, consequences of COVID. So I can basically remind you that COVID is different from influenza in that there is far more fibrosis in COVID than there is in influenza. So you might assume that there could be late stage effects. And if that happens, then definitely we need therapeutics to prevent those.
Be asked the same question for Cameron. I think you want to say where you think.
I think it's driven by the unmet medical need. I think we've looked at this in a perhaps unusual way. So perhaps I could offer a different way of looking at it. If, for example, we talk about community delivered prophylactic therapy, for example, oral antivirals. If you want to create a situation to see a resistant strain, give people an oral antiviral monotherapy and just see how long before you get one, right? With oseltamivir, it took, I don't know, next to no time. I think that's an absolutely ludicrous approach, because we're just going to see, we put limited selective pressure on this virus so far, limited, right? We're not at the beginning of the end. We're not even at the end of the beginning. And I'm very concerned that we have ill thought through policy, which is not going to be borne out by the scientific evidence. I think the other piece is affordability. So thinking about neutralizing antibodies, certainly for high-risk patients, perhaps in institutional settings, but you're giving 8 grams of protein, and the numbers needed to treat are in the high double digits. That's not really very cost effective. It also drains manufacturing capacity globally. And so while these are all interesting approaches, I think we need to be pretty selective as to which ones receive extensive funding, right? Again, we're all small companies. Some of the grants that have been made to the companies which are all in the global top 10, they've received billions of dollars. They don't need billions of dollars. They got plenty. So I think, again, a more thoughtful, holistic approach to this long term. This is not the last pandemic. No government has covered itself in glory in terms of planning for this pandemic. And I'm concerned that we are doomed to repeat the mistakes that have been evident to everybody for the next one.
Understood it. We're sort of getting through quite a bit now. I wanted to maybe move on a little bit and talk about long COVID and whether there is any applications or any of the therapeutic applications to treat long COVID. I know there are studies ongoing in that space, and there's been some talk about the vaccines having potential efficacy there. But I'll ask any member of the panel that wants to specifically comment on the potential for their therapies maybe to work on long COVID. If not, are there others that you're aware of, and is there any excitement in the space?
I think it's quite a difficult thing to study. When we were designing our phase 3 trial roughly this time last year, we knew we wanted to study long COVID. We've put several instruments into a day 90 visit after treatment started, and we have to hope that we're measuring things which are relevant to what we now understand long COVID to be. And our general feeling is that if we are able to handle the virus better in treated patients, then logically, and that's all we've got to go on, logically we'd hope to see a lower long COVID burden in those people who've had drug. But time will tell. I think, as with this virus, we know so little about it, and it's one of those things we need to watch. It's a massive burden, obviously.
Yeah, I mean, that's one of the interesting aspects of whether you treat late or early, isn't it? Is that a lot of the people who have had long COVID have never even been to a hospital. And therefore, by treating just the very severe patients in hospital, do you then store up a group of patients with long COVID? And I guess we don't know what the consequences of all these long COVID patients will be on society in the longer term. Cameron, you seem...
Yeah, I think we have some clues there. So, look, the best way of dealing with long COVID is not to get the infection in the first place. So vaccines remain, in our view, the first line of defense. There is evidence, there's a paper in Lancet about 3 or 4 months ago, looking at over 250,000 patient records, and the incidence of neurologic and psychiatric conditions was approximately half the patients, and half of those were new episodes rather than pre-existing. So there's a burden there. We also know that longer-term respiratory impairment, and some of the data is just coming through, is worsened in patients with severe disease, particularly those people who have had to be ventilated. I guess it seems somewhat obvious, but if we can prevent those pretty serious outcomes, then it maybe we can have an impact on long COVID. But I just think we're only beginning to see the longer term sequelae, and that's going to have a pretty dramatic impact in terms of healthcare resource utilization going forward.
Now move on to the variant question and how therapeutics can help, because obviously you talked about the virus. I think it was Cameron, you made the point that the virus hasn't even been put on much selective pressure really, and the variants that we know today have been generated in conditions that haven't really had vaccines. Now we've got vaccines. Do you think this is going to add to the need for therapeutics, in that we're now putting selective pressure on the virus to evade the vaccines? And
Will I go first, maybe?
Yeah, Torsten,
yeah, sure.
Thank you.
I think, I'm a virologist by training. And so first I would say it's a stupid RNA virus. So there is no proofreading. And so it will simply continue to mutate. That's the nature of it, right? And then on the mutation frequency of the virus, we put the evolutionary pressure. And that's what we see. And so I believe we need to be flexible on both fronts. We need to update our vaccines on a regular basis. And we see already the mutation hotspots impacting also antibodies, specifically the ones which are binding in the upper part of the RBD, the receptor binding domain. That's just biology, right? These are the areas of the spike protein which are the most immunogenic epitopes. They are selected on one hand by the human immune system, targeted, and they are also targeted by the vaccines. And so there is a problem coming, and that's why we need to modify our vaccines. And I think the same applies for therapeutics. I think all of the nanobodies, antibodies, whatever it is, have an Achilles heel, where if you really have bad luck, you are hit. We've seen that with the Lilly antibodies. And so I think we'll need to keep our flexibility there.
And Sander, you're obviously approaching from the native human target. So is that a way of avoiding variants from a therapeutic perspective, if I'm understanding you correctly?
Yeah, the target of the drug developed by Covercept is a host enzyme, and I would say extremely likely to mutate under treatment. So that I think answers that question. And so it could be that the virus actually mutates and evades, of course, the primary effects of the treatment. But those include, as I said, heparin sulfate proteoglycans, which are a common way of RNA viruses to get in, but also viral replication, and then last but not least, the long pathology. That's the inflammatory part, so the cytokine release, but also the fibrotic genes. So it nothing would surprise me. I'm not a virologist, I'm an internist, but my wife is a pediatric infectious disease person who has studied COVID or coronaviruses way before COVID. And when this whole thing started, she told me, okay, Sander, you can think it is going away, but I tell you, this is not going away. She said it from the start, So that's why I was interested in this particular asset, because I was thinking along these lines. And so, everything can happen, but I think when you think about type 1 interferons, or you think about these sort of mechanisms, then of course it's not so very likely that anything will happen there.
Right. And maybe on the last few minutes that we have, it's be interested to know. I mean, you're kind of alluding to I was going to ask about the post-COVID world, but it sounds like from the panel here that this is really here to stay, and post-COVID world is maybe it'd be like asking when's the post-flu world. It's kind of not a real question. Is that how you guys see it? Kind of interested in different views.
Absolutely.
Go on, Richard.
I think when we start to think about variants, we have to see how they evolve. I mean, for our drug, we're switching on perfectly intact pathways, which the virus is preventing from being activated. So if those pathways can be activated, we see strong antiviral activity in vitro, for example. So we're just putting something into the lungs which should be there. But I think, we've got to think about this viral event as a warning. It has been a disaster for the world, but it could have been so much worse. If SARS-CoV-1 had the infectivity rate of this virus, then that would have been an absolute disaster. So the rate that patients get hospitalized with this virus is 5%, give or take, and mortality is somewhere around the single-digit percentages, low single-digit percentages. So we've been quite lucky. It's been a disaster, but it could have been so much worse. And we have to hope that some really good lessons are learned, whether it's accelerating vaccine development, helping the antibody companies bring their products to the market more rapidly, and hope that there's some broad spectrum antivirals that emerge from this to get us ready for the next one.
Perhaps that's a nice segue into in the final kind of question. I'd like each panel member to answer is what are the lessons learned then? Maybe starting with Sander, what have we learned from COVID, and maybe what haven't we learned?
Well, I also had discussions with in the EU and in the European Union and in the Netherlands about pandemics before this happened. And the attention to pandemics and then the money spent on it was almost nothing. Nobody was interested. So that at least has changed. So I think, and we hope that will be sustained, because people tend to forget very rapidly. But the good thing coming out of this is that it's very likely, in my view, that there will be a couple of drugs that will be useful for other pandemics as well. And I think, the next thing that people need to understand, because there still is the idea that at some point in time it's going away. I think politicians and people who plan for the future need to understand that this is not going away. And the other thing that I think should be a lesson here is that there might be a next pandemic. People have just isolated, SARS-CoV-2 variants from Malaysia that are just one amino acid from what we now have in humans. So there's not a lot of mutations that need to happen there. In fact, in this case, from the Malaysian variant, there's not any mutation needs to happen. So there's not even an intermediary animal. It can spread directly, from those bats to humans. So, this is a lesson. This is going to happen. It's going to continue, and we just need to be prepared.
Cameron, your thoughts?
So when people perhaps suggest it's going away, where exactly do they think it's going to go to? But it's not about to hop on a spaceship and go to Mars and say, my work here is done. We're living with this thing forever, and it's doing what viruses do. So it's a little surprising that we think that we're smarter than the virus. I think the converse is true, at least right now. So my concerns are around complacency, whether that's with respect to the next pandemic or letting down our guard right now. In the United States on Friday, we have a bit of a lag with some of these metrics. There were 2,177 deaths. So that is 3 people that are dying every 2 minutes in the United States at the tail end of the summer when we're trying to vaccinate a bunch of people. So it's not done. The other thing that I think it is teaching, this whole pandemic is teaching us, is the inequities in terms of healthcare access. And that certainly is a big topic that we don't have time to get into.
Right.
But that is something we absolutely have to pay more attention to.
Okay, we've got about another minute left, so maybe Richard and Torsten, what's as briefly as you can. Torsten, go first. You're the virologist amongst us.
I really hope it's a wake-up call for infectious diseases, and so we have seen in the influenza pandemic 10 years ago that it was a complete disaster. So nobody was interested in any pandemic preparedness anymore, and I really, really hope that it's a different severity of the disease this time, of course, but that people really continue to think about it, to be concerned, and to invest both in therapeutics and in vaccines in the future.
Right, and Richard, any last comments?
Well, I think I was allowed to conclude because that's what I did in the last question, but I think we've learned a lot. I think there are political lessons to be learned. I don't think collectively we've done a bad job. It's been a misery, and the world has hurt, and hospitals have hurt, and people who work in hospitals have endured immense pain around this. But I think the industry on the whole has done extremely well and has risen to the challenge. And I think we will come out with some things which will make us much more ready for the next one.
It sounds like there are some definite positives. So unfortunately, we're now out of time. So I'd like to thank the panel for their contributions today to the debate, and wish you all the best with your endeavours and bringing new therapeutics to market for COVID, because it sounds like you're all agreed it's here to stay. So thank you very much for your time, and hopefully we can welcome you again to another panel at the SACS. Thank you.
Thank you, Max.
Thanks, Max.